Let me. So people should get comfortable with loud sounds from now. That's a heads up. It'll be fun later. You can all blame me, it's okay. Before we formally get started, welcome, please join us. We're just getting settled. Thanks for coming, everyone. We have a good group on Zoom today and in the room. First of all, I'm gonna pass on Dr. Rob McCollum. Apologies for not being here in person today. He really wanted to welcome you, Professor Melgert, and everyone to our center seminar series. Professor McConnell is the director of our P30 Center, as you all know, I'm sure, along with Dr. Lida Chatti, who's the deputy director of our center. So welcome, everyone. It is also my great pleasure to introduce to you Professor Barbro Melgert. Professor Melgert is a professor of respiratory immunology at the University of Groningen. I get that right, I think. Her group investigates how the immune system in the lung is impaired by toxic exposures and can be harnessed to treat chronic respiratory diseases like asthma, COPD, and lung fibrosis. Another important cause of aggravation of lung disease that she and her group focus on is exposure to air pollutants. And as you know, our indoor environment contains many of those types of air pollutants, including the recently described novel concern of microplastics. Funded by the Zon MW consortium called Momentum, Professor Melgert, hi, welcome. She works on elucidating the effects of inhalable microplastics on human lung health, with again a special focus on interactions between structural lung cells and the local immune system. So I promise you it's going to be a very fun talk. Before I let Professor Melgert have the stage, I just want to remind you all that we will have lunch right after this. So please stay and join us and enjoy lunch with our guests today. And then we have a 2 p.m. meeting on microplastics and microfibers as novel environmental exposures. A 3 p.m. meeting more focused on environmental respiratory diseases. A 4 p.m. meeting more focused on lung cancer and environment. Feel free to pick and choose. You're welcome to come to all of these. It's going to be upstairs in 222 or on the Zoom link that we have sent out an invite. So if you're not sure, just ask me or Dominica and we'll point it out. And I know Ryan's going to get us set up here to start. Thank you so much. And please welcome me. Join me in welcoming Professor Melgert.
Well, thank you very much for inviting me and being here first time in LA. I'm overwhelmed by all the friendliness and so Barbro Melgert from the Netherlands. And I will be talking about microplastics. I'm a translational researcher. So I understood that the audience was a bit mixed. So if I'm skipping over something which is unclear because it's not your field, feel free to ask questions while I'm talking, right? Just raise your hand and let me know. Because I think it would be a shame if you miss the story, but I try to be as inclusive as possible. Right, and what I want to talk about is, well, our studies basically, looking into the health effects of microplastics. And for that, we and others started using organoids, lung organoids, these novel 3D structures that resemble lung tissue more than a 2D culture in a dish would. It's a bit more complex, but not as complex as an experimental animal or a full human. Right, so plastic. So you might know the Aqua song, Barbie, in which one of the lines is "Life in plastic, it's fantastic." And I guess we can all agree that plastic is fantastic, right? It's versatile, it is durable, and it's cheap. So that allows us to consume lots of plastics in multiple ways. It's in everything. But unfortunately, we're not using it in a very sustainable way. So 90% of all the plastics produced end up in the environment. And I guess these pictures are just snapshots of what that looks like. So it's not nice to be confronted with all this rubbish. But the bigger problem is that this rubbish sort of degrades into tiny microplastic particles and even more small nanoplastics. So what are microplastics? Microplastics are degradation products of larger pieces of plastic or specifically produced small pieces of plastic, like for instance in personal care products, that are between the sizes of 100 nanometers and 5 millimeters. So it's a huge size range. And because they're usually made or they start to become apparent because of degradation, they come in many different shapes, sizes, colors, and surfaces. And all these characteristics can have health implications. So I'm a big Dr. Who fan, and I was pleasantly surprised that a couple of years ago there was an episode on microplastics. So we had just had a discussion about the word awareness, but I think this helps to make people aware of the problem of microplastics. It increases their knowledge.
Microplastics, alien bacteria, homing in on microplastics because humans are full of them.
Excuse me, I'm not full of plastic. Full of something.
I'm afraid you are, Graham. It's in the air, it's in your food, it's in your water. Humans have flooded this planet with plastics that can't be fully broken down, so much so that you're ingesting microparticles whether you know it or not. You're poisoning yourselves as well as your planet.
Well, I can't say it more succinctly. Everything she says here is true, right? It's found everywhere on the Himalaya, in the Mariana Trough, everywhere. And it's so small, it can end up in our bodies. And I'll take you through the data showing what we know this far about us being exposed to microplastics. So being a lung researcher, of course, I focus on the lungs. And for the lungs, the most important microplastic contamination route, of course, is the air. And we know that there's outdoor and indoor levels of microplastics. And the indoor levels are much higher. So indoor, the different studies have shown a fallout of up to 10,000 particles per square meter per day, while outdoor it's a little bit less. And that is multiple types of origins, textiles, cars, agricultural plastics, packaging. And whether these particles end up in our lungs, of course, depends on size. I don't know how many people here are familiar with lungs. Yeah, pretty familiar. So you know that anything below 100 micrometers we can inhale, but it gets stuck in the higher airways. If you want to target drugs to the lower airways, then you want your particle size to be around 2.5 micrometers. And anything smaller can end up also very deep, but can also be exhaled again because it stays suspended in the air. So microplastics are in this whole size range, so they could end up in our nose, in our trachea, airways, alveoli. But do they actually end up there? And there have been that many studies looking at this yet, but there are 3 studies that looked at microplastics in human lung tissue, and they all find fibers, particles of different shapes, sizes, and materials. But the common theme in these studies is that the most common plastic is polyester. Polyester is the most important constituent of our clothes these days. So this is probably because of indoor exposure to the textiles that we use. Yes.
It seems pretty hard to identify. Are there antibodies or something to light it up?
Oh, You put your finger on the sore spot. It's very difficult to find these plastics because there are no antibodies. There are special techniques that can identify plastics, but the techniques don't have a high resolution. So it's 10 micrometers is possible, but anything smaller is really difficult. So there is data about the larger pieces or fibers or particles, but there's very little data about nanoplastics. And of course, those are of most concern because they can go through cells, but it's just too difficult to find those. So there's this gap in knowledge about that size range.
Nobody has tried to make antibodies against it. I know, maybe it's a stupid question.
Not that I'm aware of, but I think that if anybody has antibodies, then they have a gold mine because people are looking for ways to find plastic. And now it's very, and the other problem, looking at this is that there's so much plastic in the air that you need to have really good blanks to exclude the possibility that there's stuff falling on your sample. So that is really hindering the field. Right, so I found this actually on a US website showing what they think the inhalation of microplastics is over a year and a lifetime, minimum and maximum, and they've sort of graphed it as height of a certain thing. So apparently you inhale about 2 giraffes of microplastics per year, and it could go up to the amount of Snowdon Mountain. Oh, I guess it was a UK study. So potentially we're exposed to a lot of microplastics. And there are 3 issues with inhaling microplastics. So part of it is particle related, but also part of it is additive related because plastic contain a lot of additives to improve the properties of the plastics, and these additives can leach out and cause effects. In addition, microplastic can absorb things from the environment, think pesticides, etc. That could also be a concern. So if we look at particle and fiber toxicity, I think that has been researched quite intensively because we're exposed to particles all the time. We know a lot about asbestos, silica, wood dust, and we know just inhaling lots of particles is not good for you. It leads to frustrated phagocytosis, oxidative stress, and inflammation in the lungs. And I don't see microplastics being very different in this respect. And most of the studies actually looking at the effects of microplastics focus on particle toxicity. So they look at size and shape and zeta potential and how that leads to certain outcomes. The thing I think is most important is actually the additives, because there's so much stuff in plastic that can leach out. We know that so far about 13,000 different chemicals have been identified in plastics, and only 7,000 of those have been tested for their health effects. And half of those were of concern. So think about bisphenols, the PFAS, phthalates, they all are associated with health related problems. And then, of course, the pollutant carrying, not a lot has been researched in that respect for inhalation exposure. I mean, you could think of pesticides adhering to microplastics or microbes, viruses, but I don't know of any studies that have looked at this in the context of lung exposure yet. So when we think about the lungs, of course, our lungs have been around for a while, so they know how to defend themselves against all these inhalations. And for the rest of the study, or the talk, I want to explain a little bit about the lungs and the defense mechanisms to make sure that everybody understands the rest of the steps. So our lungs consist basically of airways transporting the airs and the alveoli that are the respiratory units. So the airways have specific epithelial cells that keep out stuff that we breathe in. And they have epithelial cells with cilia, these hairy cells here that can remove particles that we breathe in that are caught in the mucus. And the respiratory units have macrophages, immune cells that can eat up the particles before they enter the rest of the body. So I want to talk about the effects of microplastics on epithelial cells and a little bit on the immune cells. And of course, ideally you would want to combine that, but we haven't gone that far yet. So what we use for our studies into epithelial cells are the lung organoids. And you basically take out the epithelial cells from lung tissue, put them in a dish in a gel-like material with growth factors and blah, blah, blah. And then they develop into these structures that either resemble airways or alveoli. So this is what an airway looks like in a dish, and this an alveolus. So big bubbles versus these sort of great black structures.
Human or mouse?
These are actually mouse, but we see the same thing. And I have an example of human later on as well. So how do we do this? We take a piece of lung tissue, either from humans or from mice. We dissociate all the cells, and then we select specifically for the epithelial cells with antibodies. And then we put these epithelial cells together with fibroblasts into matrigel, and then they grow into these transval systems into organoids. And that takes about 14 to 21 days. And the fibroblasts are needed as a sort of support cells. They make a lot of important growth factors. But we also have systems where we don't add the fibroblasts, and these usually specifically grow out to airway organoids and not alveolar organoids. And why does this work? That's because if you look at the airways, then we have two cell types, both basal cells and club cells, that have stem cell-like properties. So there always be in this whole mix of epithelial cells that you isolate, there will always be these cells with stem-like cell characteristics. And these in the dish will grow out into these organoids containing all the other different epithelial cells. So they can differentiate into goblet cells that produce mucus and ciliated cells that can move the mucus around. And the same is true for alveolar organoids. They're both the club cells and the type 2 cells have stem cell-like properties. That means that they can grow into clumps of cells and then differentiate into type 1 cells and these alveolar-like structures. So what does it look like in real life? Well, like this. So this is the mouse. You see these big bubbles, which consist of airway epithelial cells, and these dark, smaller structures that are alveolar organoids. And here for the human, again, big bubbles, and it's a little bit more clear here that the alveolar ones have all these tiny little grapes. So there are three ways of impacting the lungs. And so what is known about microplastics already, right? So what do we know about outdoor air? Colleagues of mine did a study in the Netherlands looking at the outdoor microplastic content of the air. And they found that actually most of the particles present were from tires, car tires, but packaging was also a major contributor. But this is Dutch air. might be different here, I don't know, but seeing how many cars are around here, how many roads, I'm assuming that it will be at least that much as well. And what do we know of these tire particles? Well, from epidemiology, we know particles are harmful, but it's very difficult to disentangle specific microplastic particles from other traffic-related air pollutants. And I guess Rima can tell you a lot about that. There have been a few in vivo and in vitro studies looking at tire particles specifically, but most of them focus on acute toxicity, which of course is not very the way we are being exposed, because we're exposed constantly to these particles. And most of the focus has been on particle-related toxicity, and haven't seen any studies looking at the effects of leaching chemicals from these particles. And I want to highlight this study because they used airway organoids to look at the health effects. So they got lung tissue from humans and they developed them into these airway organoids. And then after the organoids were developed, they exposed them to tire wear particles that they made themselves by grinding down tires. And they assessed the typical particle toxicity parameters: oxidative stress, cell viability, cell activation, and differentiation. And what they show, this is one of the figures from the paper, is that there is oxidative stress, there is a decrease in cell viability, and there is activation of inflammatory pathways. But this was just very short-term exposure. It was 48 hours of exposure, and they didn't look to see if this is purely particle related or whether leaching chemicals from these particles were also contributing to the effect, which I thought was a downside of the study, because I would be really interested to see what the effects of the leaching chemicals is. And one thing that is a problem for all microplastic studies is the fact that there are no standardized particles. We have standardized polystyrene, but that is not an environmental plastic, right? It's made in the lab or in the company, and it's not what we're exposed to outside. So everybody makes their own environmental plastics, so it makes it very difficult to compare studies. And I think that's where our consortium comes in, the Momentum Consortium, because all of us use the same particles. They've been made centrally, distributed to all the partners, and we all work with the same particles in the same concentrations in all our different systems related to gut, lung, placenta. So we'll be able to compare all these studies at some point. So what do we know about microplastics in indoor air? Well, most of them are of textile origin. If you look at just the average American house, it collects around 20 kilos of dust per year, and 30% of that is synthetic. So that means around 6 kilos of microplastic dust in your house. And if you couple that to the fact that, at least in Holland, colder country, people spend 90% of their time indoors, and that is a lot of exposure time. So that would explain why most of the plastic found in the lung is polyester, I would say. What do we know about health effects of textile microplastics? Well, we have some data from occupational exposures, from people in textile industries, in the nylon flocking industry. And so these are older studies, I guess, before people wore protection. So 15 to 40% of the people developed respiratory symptoms, which I found shockingly high. And that was airway inflammation and interstitial inflammation. So basically all of the lung was affected. And these people also have a higher chance of developing lung cancer. So I'm particularly interested in indoor air quality. So I wanted to look at these textile microplastics a bit more. There wasn't a lot out there when we started the studies. So we wanted to know, are microplastics harmful to the lung immune cells, to the lung macrophages? Are they harmful to the epithelial cells? And what are the effects of long-term exposure, as far as we can do that in the lab? And so we made our own fibers because there was nothing out there. And we used nylon, polyamide 6.6 and polyester. We were a bit limited in the size because the smallest nylon and polyester we could find were 10 micrometers in diameter and 15 micrometers in diameter, and we wanted to make fibers. So fibers are defined as something which is at least 3 times longer than its wide. So these nylon fibers are 30 by 10 micrometers, and these are 45 by 15 micrometers, which is inhalable, but not respirable. So they probably get stuck, if you would inhale them, get stuck in your larger airways. So first of all, what do these fibers do with macrophages? And well, macrophages are our first line of defense, and they eat whatever they find that they think doesn't belong in the lung. And I guess most of you will have seen pictures of macrophages filled with carbon particles. That's just what they do. If they can't digest it, they sort of put it somewhere in the corner of the cell and hope it just stays there and doesn't affect them too much. So will the same happen with microplastics? Okay, so sound alert. Before I start the movie, the green blobs here are macrophages, and the fibers were dyed pink. And we put them together wondering, okay, so what is going to happen? These macrophages live on plastic, right? So why would they start attacking a plastic fiber? If they would. So. So, Kirsten, my daughter, who put the music on there. But they really go and try to do something with it, and they even seem to be fighting over fibers,
So
right?
what kind of macrophage you are using? It's a raw?
No, these are actually alveolar-like macrophages, so they're primary cells. So alveolar macrophages derive from fetal monocytes. So we have these fetal monocytes that we can propagate into alveolar macrophages. So they're very young alveolar macrophages. So this is over a night. So what happens when they're exposed for 24 hours? Well, they are taken up in the cell, and I am surprised that they're even, I mean, these are quite big fibers, right? I mean, a cell, a normal alveolar macrophage is around 15 micrometers. And this one here has 3 of them inside. So we're now investigating a little bit more what is happening with these macrophages. So there is indeed increases in oxidative stress, and they make inflammatory cytokines, but not the ones you would expect, not the classical TNF alpha IL-1 beta ones, but they make chemokines that specifically invite neutrophils. So there seems to be some cooperation between the two, like I've eaten this, I can't handle it, I need somebody else to come and help me with these fibers. So that is currently what we're investigating.
Where are the macrophages start to go? I mean, if they just die in situ, then the problem not solved, right? So do they?
Exactly. So what I think would probably happen in vivo is that you get granulomas, because one macrophage eats the fibers, dies, then another macrophage will come and eat that dying macrophage, encounters these fibers, cannot handle them. So it's the same as with tuberculosis, where they can't deal with the threat and they start walling off. So I think that would probably happen, but I have no evidence.
They don't, yeah, they don't go somewhere where they can be secreted by the body.
Yeah, I guess some of them will be taken up with the mucociliary elevator and then be swallowed, and then they end up in your gut. But we don't actually know. We've treated mice with the plastic fibers, and we do see that within 1 week, the number of fibers clearly becomes less. But where they go, I don't know. So moving back to epithelial cells again, of course, that's the other first line of defense that needs to deal with these inhalable threats. And we also use this organoid model, but our model was slightly different because we add the fibroblasts to the culture, and which means that they can grow out into airway and alveolar organoids. And because we wanted to look at long-term exposure, we added the fibers from the start of the culture. So we added the cells and the fibers at the same time and then see what would happen to the development of these structures. So to remind you, this is what they look like in real life. And this is the cultures with the fibers. So here we have the control situation with the large bubbles being the airways, the smaller, darker alveolar structures. And you can see with polyester and nylon that there are, especially with nylon, dramatic changes. Polyester, there are some changes, but with nylon, there is a complete lack of these airway organoids. They don't seem to be growing anymore. So we quantified that, but I just wanted to show you what this looks like. So I made a movie of 14 days culture. And it's like 30 seconds a movie, but it took so much time because you have to make pictures of your culture of the exact same spot multiple times a day. So I really want to thank the postdoc who actually did this for me as a favor, because I think it really shows the difference between the two conditions. So left for you is the control condition, right is the nylon exposed condition. So you clearly see that in both conditions, organoids start developing, but the nylon exposed somehow lag. There are some airways developing there, but not as many and not as big. And if you quantify that, it becomes really clear that nylon really inhibits this outgrowth of the airway organoids, while the effect on the alveolar organoids doesn't seem to be there. Polyester also does something, but it's very mild compared to nylon. So of course we focused on nylon then. And we did ask, is it the particles or is it something leaching from the fibers? And the way we addressed that was we had the normal condition where we had the fibers inside the gel together with the cells. We also added the fibers on top of this gel just to prevent them having direct contact with the cells. And we made a fiber wash, just getting the leachate out of the fibers and then adding only that leachate to the cultures. And the results were quite clear, luckily. This was the normal condition with the fibers inside the gel, and this was fiber on gel, and this was the nylon wash, and you see the exact same thing happening. So it's not the fibers, it's something leaching from the fibers. Well, you'd think with all the analytical techniques we have these days, that would be easy to find out what is leaching from plastic, right? We still don't know. We found cyclic oligomers that develop during the production of nylon. So we isolated those from the leachate and tested them. No effect. We found PEC 600, tested that, no effect. And then we started to test all these compounds that have been found in nylon before, bisphenols, PFAS, benzophenones. Yes.
When you wash the nylon, was it with like just water or complete media or?
We washed it in water and added a little bit of that to the cultures. We also did a dose response, and we even tried to just strip the particles of anything by treating them with hexane and then just add the particles, the stripped particles. But if you strip particles of all the additives, then they aggregate, and then you can't deal with them anymore. So that experiment didn't work out. So it was water. Anyway, so we don't know what is causing this effect of nylon. And so the most obvious candidates were the culprit. So that probably means that whatever is causing this effect is present in very low concentrations and has a very dramatic effect, which I find a bit scary.
Could you figure it out by looking at gene expression?
We'll get to that.
Okay.
Okay, so we were looking at developing organoids, right? But this other study had looked at developed organoids and saw these effects. We thought, no, let's give that a try too, right? Because sitting as we sit here, we all have developed lungs. So how would that affect us? And the way we did that, we had our organoids developed for 14 days, and then we added the fibers on the gel, or we added the fiber leachate, and then had them sit around for another 21 days. So this is a bit difficult one. So we had control condition with nothing added, grown for 14 days or 21 days. This is the conditions where the nylon was inside our positive control for either 14 days or 21 days. And you see reduction of the number of airway organoids. And this is when we added the nylon on top, or we added the leachate starting at day 14. And there we don't see a lot happening anymore, which is good news for us. That means that it's not directly toxic. It's just somehow changing the developmental pathways of the epithelial cells. So then we go to gene expression. Because what do you do when you try to find out how things are changed? You're chasing a mechanism, you go on a fishing expedition and you do RNA-seq analysis these days. So what we did was we cultured the epithelial cells in the fibroblasts with nylon for 7 days. We wanted to look at the early changes in the epithelial cells. Then we specifically isolated the epithelial cells out and the fibroblasts, and then did RNA-seq analysis on the two fractions to see what was changing. I want to say there were a lot of genes changing. So I'd say by accident, we hit upon this family of genes, the HOX genes. And these genes are very important for cell fate decisions. And they have been shown to play a role in embryogenesis and also in lung development. And specifically, this gene HOXA5 was highly upregulated by nylon treatment. And there was this one publication that showed that HOXA5 was involved in airway epithelial differentiation. And the way this was told in the paper was that you have these basal cells and club cells that can become goblet cells and ciliated cells, but only if there's no HOXA5 present. If HOXA5 is present, then these do not develop anymore. And then you get these sort of aberrant airway organoids, very small, non-differentiated organoids. So what we thought were alveolar organoids were actually these aberrant airway organoids with EMT features, so sort of a precancerous lesions. So then we wondered, so if we inhibit this gene or this protein, HOXA5, do we get our phenotype back? Do we get our airways back? And that's what we did here. We treated the organoids with this inhibitor of HOXA5 called LE135. And if you do that, you get your airways back. So we were very lucky here that this one gene apparently was really important for inhibiting the airway growth. And that is induced by nylon. But it didn't get us any closer to finding out what is in nylon, unfortunately. Yeah, so we are looking now, actually we're trying an expression system with HOXA5, so we can quickly scan lots of compounds to see which one induces HOXA5 of a reporter system, I should say. Because doing these organoid, it's 40 days, right? It's a lot of money, takes a long time, so you can't do that with many compounds. So if we have a reporter system, we might be able to test more compounds. All 7,000 of the ones that are known.
Sorry,
Yeah.
if you use like, immortalized human bronchial epithelial cells for your reporter, that work?
Yeah, that's what we tried. The only problem is that these immortalized cells already have higher expression of HOXA5. And that's probably most of them are cancerous, right? So that's probably part of
Well,
the phenotype.
The immortalized are not all cancer.
Yeah, so we tried with the A549s.
Yeah, but those are cancer.
Yes, so that didn't work. So now we're trying the CALUs.
Also cancer.
Yeah,
But they have human bronchial epithelial cells that are immortalized, that are not cancers.
the
HVACs.
the HVACs, yeah.
And then BAS2Bs, but
Oh, yes, and we're also trying BS2Bs. So yeah, we were trying multiple, all the epithelial cells we have in our lab. But it's been proven very difficult to set up this reporter system. I don't know, maybe we're just unlucky with this gene. So there seems to be a correlation also whether the cell's cancerous or not. So I'm really interested also to see an interaction with lung cancer development. So for the session later this afternoon on lung cancer. So to wrap up, microplastics are a novel type of air pollution, and we have both indoor and outdoor types. And I think the studies do show that they, in high enough concentration, are harmful to lung organoids, both mature and developing organoids. So for nylon, we know a little bit more now that it's chemicals leaching from the nylon, and that it's increased HOXA5 expression responsible for the effect. So what could be the implications of this? So for the nylon, I would say that developing or repairing airways might be at risk for the effects of nylon. So think children, but also people with lung disease that are dependent on repair, because that is what this organoid model models. But of course, we need to know what are harmful concentrations, what is harmful exposure. And for that, we need to know how much is in the air here and how much you breathe in. I myself think let's ventilate as much as I can because that will keep levels down. And I got a bit worried about this. I saw this by accident on the internet, this American company promoting nylon carpets with children playing on them, breathing in these fibers. So no, let's not do that. I wouldn't get nylon carpets in my house, I would say. So what are the key open questions in the field? We've touched upon those already. So what is the actual exposure of humans? Which plastics are most commonly found? And I think most importantly, which chemicals leach from plastics? And then once we have that information, what is the most important for risk assessment? Particle or chemical toxicity or the combination? And this really put me off recycling. Because with recycling, we're putting together all these plastics from different origins with different unknown chemicals. We're making the mix even more complex. So I think recycling actually is not a good idea. We should just reduce our plastic use. And with that, I would like to thank many people who worked on these studies, especially the 3 postdocs, Francine, Shanshan, and Glenda, who did excellent work on getting this published in the Blue Journal, but of course also the Dutch government for having the vision to fund microplastics research, because I think Netherlands is a good place to study microplastics at the moment. So, end of the talk. I'm happy to take questions. Yes.
So given what you're saying about the development on the airways, I would be very concerned about pregnant women and their exposures. So is anyone studying that aspect?
Well, within the consortium, we have people looking at development, so they're looking at placenta and the in clinical cohorts also the children. But we haven't got a very good method yet to measure nylon in blood, because that's something you would need, right? Is if you can measure nylon in blood of pregnant women, you can maybe do associations. But I totally agree with you. As a pregnant woman, I wouldn't fly on that carpet. Yes?
I was curious, I'm also wondering about seamstress. There's a lot of, especially, well, my mother-in-law, that was her only job since for a long time, and when she immigrated, she get doing that type of work, and so I'm wondering, for people that have worked that occupation for so long, and you mentioned that, but I'm wondering if there's, as they get older, if they're having any, if you're seeing any other side effects.
Well, probably, yeah, I would probably think so. Of course, there's no data. And of course, I think the amount of synthetic material that we're working with is increasing. I mean, when I was younger, most of our clothes were cotton, wool, etc. There wasn't hardly any synthetic clothes. But look at my kids now. It's all synthetic junk that they buy because it's cheap. So I guess these studies will have to be done, and will be coming out at some point. But I would worry indeed about people working with synthetic fabrics. People working in the factories producing these fabrics, but also 3D printing, which uses plastic. There's also lots of dust there. So I think there are all these old occupations that might get these new exposures. So I think it would be really great if we could have this scale of occupations with high versus low exposure, and then we could use them in the studies that are already around to see if they're associations with certain occupations. Yes.
So I have so on that slide, you mentioned that the particles per square meter. So is that the reason it measures the position rather than the air? Should be cubic.
Yeah, so it depends. So there are two ways of measuring, as I understand it from the literature.
I believe Professor Melgert showed the emission rates, how much it emitted from or into indoor air and outdoor air, and that would be particles per square area per day. That's different than like a concentration you might measure in the air, which I believe we were chatting before. That's so very challenging to kind of quantify them appropriately. And
Yeah.
Is that your question?
Yes, yeah.
So what I do know is that, for instance, in the nylon flocking industry, they measured the concentrations in the air. So there they had, so where 30 to 40% of the people develop symptoms, they had 7 milligrams per cubic meter of nylon or polyester. Yeah, exactly. But then, I mean, 40% people developing symptoms, you're not surprised.
And then what is the potential mechanism related to asthma?
Wow, that's a very good question. So what I can imagine, so for nylon, if your airways don't repair well, then of course the barrier is leaky. That means that allergens can get into through this layer much more easy, or even the microplastics could absorb allergens and be a vehicle that transports these allergens into dendritic cells that then get a alarm signal because it's a huge particle and stuff on there. So it might lead to sensitization, but there's not a lot of evidence for that yet. It's all supposition.
Yeah,
But
what
that's-
was the allergic asthma?
That's a very good question. Yeah, I don't know.
Very interesting.
Yeah. Yes.
Great talk, this is fantastic. I was just curious about the difference between the toxicity between polyester and nylon. Do you think it's something about like the way nylon leaches or the composition or
Yeah,
like what picks up?
I would love to look at that. So I really want to know what leaches out. Why can't nobody, like, we have so many chemical specialists. Why can't they just tell me what leaches out? Anyway, but I think that that's something I would like to look into. So what is the difference between polyester? And we also tried PVC, polypropylene, and they all have different effects. And I really would like to compare those. But there's just, these models take so long. So there's so little time. So yeah, there's lots of place for other people to work on this.
And sorry, just because related to what Sherry was asking, maybe it's not the water soluble stuff or like.
Could be, yeah. I don't know. We haven't had the time to look at that because we do see some effects of polyester.
Yeah.
But we haven't looked to see if that is leaching effect or the particle. Yeah. Don't know. Yeah.
I have more questions related to like maybe what's leaching, a couple of them. First, have you tried different nylon suppliers? And then, so it's.
Yeah, so we actually also used just fabric that we bought from a fabric store, nylon fabric, and tested that, and that was. It's hardly possible, but even more inhibitory than the clean nylon.
Wow.
We use nylon 6.6, but we also have nylon 6 particles. They also inhibit this. So nylon 6 is very different chemically from nylon 6.6.
The particles or just the leachate?
We haven't tried the leachate of nylon 6 yet. But the chemists who are working on the
Yeah.
leachate told me that the nylon 6.6 leachate has monomers that belong to the nylon 6 that shouldn't be there, that somehow there is some, because this is all unregulated, right? I think there's just a company somewhere mixing some nylon together. So it seems that there actually is a mix of nylon 6 and 6.6 in what we're using. And we have this other nylon 6.6 that was completely different source, different particle types. So these were roundish particles. Also had very dramatic effect. We had leachate from that, very dramatic effects. So the short answer is yes, we tried. You had a question?
Yeah, I'm just curious, the nylon and the polyester, their mechanical property the same or, I know the macrophage tend to uptake like a stiffer
Yeah.
things, so the nanoparticle regarding the
Wow.
mechanical.
I have
Property?
no idea if they have the same stiffness. What is interesting though, is that this other study that also used organoids to look at effects on organoids, they found that with polyester fibers that the epithelial cells tend to start to grow on them, like it's their
Yeah, so
substrate.
their surface charge or
Yeah.
that kind of things.
Yeah.
They have potential.
Yeah.
Different.
Yeah, so we haven't really seen that. We sometimes see an organoid that sort of grows around a fiber. And we've never actually seen that the fiber is covered with epithelial
Do
cells.
you notice that the macrophage tend to fuse, trying to?
Yes. Yeah, definitely. Because they need to fuse, otherwise they're not big enough. Yeah.
They're forming like a foreign body giant cell.
Yeah, so that's why I think that in vivo, they probably will form these granulomas with giant cells inside, but we haven't actually got the histology to prove that yet. Yes.
Technical question.
Sorry, Ita, do you mind just 1 second? I know we have 2 more questions,
Okay.
but we have one from Zoom just to let people chime in. Yes, Dongshi Chen, do you mind unmuting and asking your question?
Hey, This is
Thank
Dong
you.
Chen. Yeah, very interesting work. So I just interesting. So the microplastic can both affect the organoid and macrophage. So did you try to co-culture the organoid and macrophage to see if this kind of the microplastic induce the change of the macrophage, whether it's a pro-inflammatory or help to repair the organoid? And the other question is that because I saw the organoid culture in your hand is kind of the mix of the alveolar organoid and airway organoid. So based on your experience, so how much percentage of these two kind of organoid in the mixture?
All right,
Thank you.
first question. Yes, we are trying co-cultures. So if we co-culture the epithelial cells with macrophages, alveolar macrophages, we actually see an increase in the number of organoids developing. And if we add the nylon fibers to that, we see decrease, but that's exactly the same whether or not there are macrophages present. So the macrophages do something to the organoids, but the nylon does the same thing.
So it's worse. Sorry. So it's worse than only the microplastic treatment, right?
Yeah, well, what we haven't tried is treating the macrophages with plastics first, and then use those to put into the organoid. So that is, I guess, another experiment that we'll be doing.
Okay.
What we do know is that the macrophages, that they show all these so they eat up the particles, they show oxidative stress, make chemokines, but they do not do this to the leaches. So the macrophages really do something particle-based, but not leachate-based. And then your second question was?
Oh, the second one is a technical question. It's about because how many percentage of the alveolar and airway organoid in the mixture? Yeah.
We usually get around, let's say, 500 alveolar organoids versus 50 to 100 airway organoids. But we also have used a dedicated airway organoid model without fibroblasts, only the airways. And then, of course, we see the exact same thing happening with nylon. So it's it depends a little bit on what we use, but we consistently see this on airway organoids in all our models.
Okay, thank you.
This is Dr. Kim. Can I ask you some questions?
Of course.
This is Dr. Kim, Hwang Jin Kim. I have a couple of questions regarding the results on alveolar organoid. I don't think you touched upon that. And the other second question is there any size effect, like 100 nanometer versus 1 micrometer or above microplastics on any of your results?
So I'll start with the last question first. We tried different sizes of the nylon particles, so not the fibers, but the particles. So smaller than 1 micrometer, 1 to 5, and 5 to 10 micrometers. But it's leachate based, so they all show the same thing. Might be different when we start incorporating macrophages there. And then the first
alveolar organoids results.
Oh, yes, the alveolar organoids. Yeah, so we didn't see a huge difference there, but we think it's because we misclassified the organoids there as alveolar while they're actually non-differentiated organoids. So we're looking into that a little bit more because we think they're actually just epithelial cells undergoing EMT and just staying in this undifferentiated state. But we're currently working on that.
Okay, so one final question from me is, when you co-culture macrophage with airway or alveolar organoids, have you tried that?
Yes.
And any differences in response to microplastics?
No, we find, so there is a difference when we culture the macrophages with the organoids, we get more organoids when there are macrophages present, but the effect of nylon stays the same, whether or not there are macrophages present.
Uh-huh, that's interesting. Thank you very much.
You're welcome.
You had a question and then afterwards, one more from the back.
So just a technical question on how you made the shredded particles, is like that tumbled or?
No, no, that's a lot of handwork. So what we do is we have a spool where, and we make like, do like this. I'll explain that. And then we have this, and then we cover it with tissue tape, and then we cut blocks out of it, and we freeze it, and we cut blocks of that. So then we have like blocks like this, and then we use a cryostat to cut it at exactly 15 micrometers or 45. That's a lot of work, but you get a very defined size range in that way. So I'm very happy with the particles we get from momentum, because that is just particles, but it's a lot easier than making all these fibers.
Both of my questions were answered, actually.
Oh,
Okay. Any more questions from the room or from Zoom? Folks, you're welcome to just speak up if you have any on Zoom.
I wonder, so you tested all these compounds and didn't find the one yet, but what if it's a combination of?
We also tried that.
Oh,
We tried so many things, and it's been such a frustration that yeah, we need more chemists, I think.
Well, before we give Professor Melgert a round of applause, I just want to remind everyone, we have plenty of lunch boxes. Please grab one and stay with us so you can enjoy lunch with our speaker. And we also have a 2 p.m. meeting on just the topic of microplastics as a novel exposure, 3 p.m. on environmental respiratory disease, and 4 p.m. more focused on lung cancer as outcomes. Both on Zoom and upstairs in 222. You're welcome to all of these. Please join as much as you can. Thanks so much for coming, and thank you, Professor Melgert, for an excellent talk.
Thank you. And I've enjoyed myself. Lots of questions. I like that.
Thanks to everyone on Zoom as well. We're gonna